Harries is Professor of Molecular Genetics at the University of Exeter, College of Medicine and Health, where she leads the RNA-mediated disease mechanism group. Her group has interests in messenger RNA processing and epigenetic gene regulation in the context of aging and chronic disease. She is a co-founder and the chief scientific officer of Senisca, a xenotherapeutics company. With that, let me start the interview. Welcome to Modern Healthspan, and it's a pleasure to have you on our channel today.
Oh, thank you. It's very nice to be here.
So your lab is doing some interesting work on RNA-mediated, how diseases of aging are mediated by RNA and senescent cells. So what I'd like to really talk about is that today. Looking at RNA, so how does RNA mediate the diseases of aging? I mean, can we kind of start at that basic level?
We can. It's not actually, that's actually probably about the most complex place that you could start at. So obviously RNA underpins everything. So every gene in your body, in order to be able to be turned on and expressed and to do what it needs to do in the cell, does so via an RNA intermediate. And the particular area of RNA biology that we work on is what happens to that RNA. So your gene gets switched on, your cell makes an RNA, and we work on what happens to that RNA before it actually gets translated to make a protein, or not. Actually, some RNAs don't get translated. And it's the kind of cellular dimmer switch, if you like, on the genes, and the ability of your genes to adjust their output according to what the cell needs. So obviously your cells are constantly bombarded by things from the internal and external environment. And to keep your cells healthy, your genes need to be able to roll with the punches and to change what they're making to fit the conditions they find themselves in. And where RNA comes in respect to aging, is we know as people age, you become frail on a physical level, but you also become frail on a molecular level. So you stop being able to respond to stuff, and the processes involved in the biology of RNA are very intertwined with the molecular adaptability and plasticity of our cells.
When you're looking at the RNA, so the RNA is making the proteins, and like one piece of RNA can make multiple different proteins, right, depending upon how it's spliced.
Exactly.
So, right. So can you explain kind of what RNA splicing is, and then well, let's start with that.
Okay, so when your genes get switched on, they make what's called a pre-RNA. That pre-RNA has all sorts of stuff in it. So it has the bits of the gene that are needed to make the protein, or whatever the RNA does. Some RNAs don't actually make proteins, but it also has a lot of other stuff in it. So it has stuff at the beginning, which is to do with helping that RNA be translated. It has stuff at the end to moderate stability. And the bits that make the proteins are usually broken up into chunks. And we used to think that one gene made one RNA, made one protein. Not true. One gene, as you correctly say, can make lots of things. So the average is about 3 or 4 different proteins, but there are genes that can make hundreds or even thousands of different RNAs from one gene. So there's a case in the fruit fly Drosophila where one gene can make 38,000 different RNAs, depending on which exons get left in and taken out. So the way I always like to explain this to my undergraduates, it's a little bit like having a recipe book. And the basic recipe is for cake, but you can make a Victoria sponge or a chocolate cake or a banana cake, depending on whether or not you put the chapter for chocolate in, right? So if you take that bit of the, they're called exons, these chunks. If you take that exon out, your cake is still cake, but it hasn't got the chocolate, so it's a different sort of cake. And it's a little bit more adaptable even than that. So quite often what we find is a gene will produce many different RNAs. Those RNAs can be turned on at different times. So some of them might be expressed in early development, whereas others might be expressed in maturity. They can be expressed in different places. So a gene might make a set of RNAs that are expressed in the brain and another set that are expressed, for example, in the liver. Or it can make sets of isoforms or different RNAs which have different jobs. So they basically address the same process, but they might do so differently. So if you've got a gene that says to the cell, determines whether or not that cell grows, you might find one set of RNAs that say grow and another set that says don't grow. So it's the balance of what's made which determines what the cell does. And that's why it's so important that as we age, our ability to make those decisions as to what gets made is maintained. And that's what happens with aging, is we lose our ability to do that.
But what do we know about why we lose our ability, and in what way do we lose it?
Okay, so this is the simple answer to that is there's still a lot of unanswered questions. The more complicated answer, so each individual exon, a bit that needs to be taken out or put in, has its own controls. So they're independent events. And the decision as to what gets made is primarily made by a class of proteins called splicing factors. And these basically stick on to the bits, round the bit that might or might not be taken out, and make that decision. Now, as we get older, the levels of those proteins that make that decision usually drop. They change. It's usually in a downward direction, but they need to be really responsive. They need to be able to be, very quickly switched on and off, so the cells can do what they need to do. As we age, it's a combination of the stimuli that the cell's having. So things like DNA damage, things like oxidative stress, all of those classical things that we know are associated with age, interfere with the machinery that communicates from the outside of the cell to the inside of the cell. And we basically identified those pathways that that regulate those splicing factors, and they get basically switched on continually and not able to be switched off, which means that the splicing factors are that they're continually getting a negative signal. But it's also compounded by the fact that these genes, they're ever so clever, they regulate themselves. So they regulate their own levels by splicing. So they put in a bit of, they put in an exon, which actually causes the RNA to be degraded rather than translated. And that is under the control of that splicing factor. So if you've got too much, it will switch itself off. So they're designed to be held in a really nice, tight band in the cell. You don't want too much and you don't want too little. So there are mechanisms which will bring them back down. But once they fall under a threshold, there's no mechanism to bring them back up. So that's what we're trying to do.
For example, if I think about mTOR, right? So, I mean, mTOR is not a splicing factor, it's a gene, right? But we look at pathways that turn mTOR off or turn it on. Yeah. So do splicing factors work in the same way, that there'd be pathways that impact it and turn them on or off?
Exactly. And I think that's one of the things that we've spent about the last 6 or 7 years doing, is to unpick those pathways, to figure out which pathways are doing it. And these things are never simple, because all of those pathways not only regulate the splicing factor genes, but they regulate a lot of other stuff. They also talk to each other. So it's really hard to dissect out the effect of one pathway from the effect of all of them, if you're with me. But we tied it down to a specific couple of pathways, although the others are probably important, but more particularly to the genes at the end of those pathways, which are the things which are directly regulating.
Okay. So what kind of things would impact these splicing factors? I mean, again, like using mTOR as an example, I mean, we know like protein or sugar or whatever, turns it on. So what kind of factors?
So the answer to that is everything. So these things are part of the battery of defenses that your cells have against change. So, not surprisingly, they are affected by all sorts of things. So, so certainly proteins, amino acids, signals, inflammation, oxidative species, cytokines, mechanical stress, cellular stress, everything. So they're a real interface between what's happening to the cell and what the genes are doing.
Okay. And yes, now, so thinking about that, so the splicing factors are going to affect every gene, right? Because all genes need to be spliced, or all RNA needs to be spliced after being.
Most of it, yeah, about 98%. About
98%. Okay. But it would affect different genes in different ways depending upon different inputs.
Yeah. So it very much depends on So the control of, for any given gene, the control of what gets made it's a specific balance of splicing factors. So not all splicing factors are in all tissues. They're all important in all tissues, but you get slightly different balance of them in different tissues. So I guess it would depend. It primarily depends on what's normally expressed in that cell, that type of cell, under what conditions. And so you might find that you've got, one particular stimulus might cause the dysregulation of a smaller group of splicing factors, which is going to impact on a specific set of transcripts. Mm-hmm. But that might be different from tissue to tissue, and almost certainly is different from tissue to tissue, which is why some tissues are more sensitive to particular kinds of stresses than others.
It changes as we age, right? It becomes more frail or less adaptable. Yeah. Are the changes predictable? I mean,
no. So, well, they probably are, but we just don't know them yet. So there's going to be a subset of transcripts which are regulated by a given splicing factor. Yeah. And we can, to a certain extent, predict from the sequences which splicing factors are likely to be regulating which transcripts. We can do that. Mm-hmm. The problem is that the binding sites on the gene, on the RNAs, for these splicing factors are short and degenerate. And by that, I mean it's not a consensus sequence. There's wobble. And so whether or not that splice site gets used depends on what's bound and how strongly it's bound. So there are two types of splicing factors. There are splicing factors which promote splicing and splicing factors which inhibit it. And if you've got a splice site and you've got the stuff bound, whether or not it gets used is dependent on whether or not you've got more activators than inhibitors. If you've got more activators that are able to bind, then that will be used. If you've got more inhibitors, then it won't. So we can predict on a kind of individual gene by gene level, splice site by splice site level. This splice site, it looks like it's possible that these might be regulated by these splicing factors. So you can sort of get some ideas, but there's enormous redundancy. So you might find that this particular splicing factor isn't where it should be, but there might be another one which will do the same job. So you can't look at the transcriptome as a whole, so the collection of messages that come off a gene, and say at any particular age, you can't predict what might be happening unless you get in there and measure it.